I'll fix it. Yeah, so we're just going to lecture this week since I missed yesterday. So we'll lecture today, tomorrow, and Friday. And then I'll video them, and then I'll put them online for you. Does that sound good? Where you guys have them? Hopefully, I bought this new microphone and it works. Right? Okay. Any questions from last time? I think I left off here, right? Might have got a little farther than this, but this is from your actual textbook right here. So this guy right here. And I like it because it actually kind of talks about how this countercurrent exchange actually works. And really, it's a countercurrent exchanger up to that point of the thick limb where it actually becomes a multiplier, right? And so remember, the big difference between an exchanger and a multiplier is a multiplier uses ATP, right? It actually pumps to really, concentrate our solute of interest here into the interstitial tissue here of the medulla. So in those pyramids, basically what the loop does is add a bunch of salts to the medulla. I mean, that's what we're doing. We're putting a bunch of solutes in the medulla. So our osmolarity is going to be really high, right? Really high. And what's really cool is that the descending limb, as you can see, is permeable to water. And because there's a lot of salt, water leaves. But what that really does is it removes some water, but it concentrates our filtrate. So it's really high in salt, right down here. And then when I then open up those sodium channels, then the sodium will move into the interstitial space because it's so high, so I can remove water efficiently, move sodium chloride into the deep medulla. And then as we move back up towards the cortex, this is where I then pump the salt out to get it really concentrated. So I have a really, really high osmolarity or lots of salt or solute in the interstitial fluid of the medulla. And what that's going to let me do is fine-tune how much water I'm going to allow escape the urine. Right? And that's really what the collecting duct's job is going to be. Because remember, as the nephron comes down, hairpin turns, comes up, the collecting duct then dives back down, right? And then down here, where we get the papilla of the medulla and our minor calyx, that's where we're going to release our urine. And by the time we get to this point up here in the distal convoluted tubule, we should be either just under 300 milliosmoles or even more as far as hypotonicity. And at this point right here, about 80 to 90% of the filtrate has been reabsorbed. Okay? And so now we can fine-tune how much of that water we need to lose at the collecting duct, and that is going to be controlled by hormones, right? And our ability of our part of our brain called the hypothalamus to kind of regulate, and the kidney itself actually, as you'll see, regulate kind of when we're going to reabsorb water and when we're going to produce dilute urine, depending on what we're doing, right? How much we're drinking and what our blood osmolarity is. So as you can see with this little figure over here, what it shows you is that kind of at the top up here where we're at 300, you'll basically have solute concentrations in both of the tubes here, or at least close, that are really high in solute in water, right? And then as you move down, then we're going to, the descending limb is going to be permeable to water, while the ascending limb is permeable to salt. And what that leads to, in effect-wise, is the pool of salt, right, into the interstitial space. And as you continually move fluid through that, like this, we will continually add solute to the medullary interstitial fluid, which will allow the collecting duct then to fine-tune water absorption. Now, the mechanism of that is pretty simple. We've already kind of talked about how this works in other places. This will be really similar to some of the gill models we looked at earlier. And what you can see is driving our removal of salt, if we're talking about the ascending limb here of the loop of Henle, is going to be this symporter right here, which you've already seen before. right? It's the sodium chloride potassium symporter. And you're using the sodium gradient to pull chloride and potassium in. And this is driven by the ATP pump over here, which keeps sodium low in the epithelium. The potassium always escapes back through a potassium leakage channel. There could be potassium leakage channels on both sides that allow potassium to leak. There's also a potassium chloride symporter here, and this is that cystic fibrosis transmembrane regulator protein that we've talked about multiple times that allows chloride to escape into the interstitial space, bringing sodium and chloride into the medulla, right, into the medullary tissues. As we move into this is the thick limb, this pump is really what drives our multiplier. Lots of pumps, lots of ATP. And the reason this looks wider is because the epithelium goes from almost a squamous epithelium to a cuboidal epithelium up here because we've packed a bunch of mitochondria into it so that we can drive these pumps. And so that's why you get like a thin limb and a thick limb of the ascending limb of the lupin. Now, what's really interesting is if you have slightly high blood pressure, one of the first things they might do, or their first line of medications, will be to block this. All right, if I block that, you see this loop diuretic. So diuretics are going to block urine formation, right? And if I block this right here, what that's going to do is prevent the reabsorption of sodium, right, and chloride from the filtrate, which means over time, if you take this medication daily, the hypertonicity of the medullary space drops, right? It's going to become more and more isotonic. Because of that, the power of the collecting duct to concentrate urine is going to drop. So you'll produce more urine, which will actually drop your blood volume and lower your blood pressure. All right, so that's usually one of the first lines of medications they like to use, or they might put the diuretic in combination with a beta blocker, which is going to block another neurotransmitter or hormone that we call epinephrine. It's mostly neurotransmitter that will actually loosen up the blood vessels and drop blood pressure. But this is where a lot of the medications that are diuretics are where they're going to work. Or if they give a person a diuretic because they have swollen legs or they have congestive heart failure or whatnot, it will block that symptom. Notice down here that we also have the help of this, right? So where does bicarbonate and these protons come from? What enzymes in here? Carbonic anhydrase, right? Good. Yeah. So they express carbonic anhydrase. Remember taking carbon dioxide water, making carbonic acid, right? Which will then break down into bicarbonate, and we use that to also help us pump sodium out, right? And then the bicarbonate, remember, we use a good buffer, isn't it? And that'll enter the bloodstream. And then we can get rid of the protons to get rid of acids, right? So that's kind of how the ascending limb works. And we can use carbonic anhydrase, again, I think I mentioned this in the last lecture, to kind of control our pH, right? If we take in a bunch of acids, we can pump out more hydrogens. We can also allow bicarbonate to escape into the filtrate if we need to, okay? This makes sense? So here's our other scenario. So this scenario here is, notice that if hormones are present, I can produce a urine that's about 1,400 milliosmoles. It's about 4 times that of blood osmolarity, right? A little over 4 times that of blood osmolarity, which means I reabsorbed a lot of water, right? Or if we go to this one, if you look at this guy here that I pulled off the internet, notice it's 65, right? And so in this instance, there's no hormone that's there, and we will produce a very dilute urine. So really, the collecting duct and the distal convoluted tubule kind of fine-tune how much urine we produce based on osmolarity in the body. So that's a good example of dilute. We're still producing, if you notice here in the interstitial fluid, 300 to 1,000, 1,200, right? Very hyperosmotic interstitial fluid, but the collecting duct simply is not permeable to water, right? And so water doesn't leave. And you can already probably guess what protein we're going to monitor here or insert into the membrane are going to be aquaporins, right? Those water channels. Right? So those water channels are what are going to kind of control water movement in the collecting duct. So the hormones that we're going to talk about here are going to help. The first one, at least, which is called ADH, is going to help cause the insertion of water channels into the collecting duct epithelium, which will allow water to escape the filtrate and concentrate the urine. Obviously, those aquaporins are absent here, right? So we produce dilute urine. The other picture I kind of went out and got for you is this one that kind of shows secretion or reabsorption across the entire nephron. So I always like to point out again, remember up here, there's nothing happening as far as secretion and absorption, right? In the glomerulus or in Bowman's capsule, what we call the renal corpuscle, because this is where filtration happens, right? And then the renal tubule, which is kind of the rest of this, of our uronephros tubules, what kind of modifies to filtrate, right? We call this definitive urine, right? And then as far as it coming out, and then this will be our primary urine down here, right? The urine we produce. So along the loop of this picture, it kind of shows you what's secreted, which will be the arrows, the normal arrows, right, that come in. And then you can also see arrows going into the filtrate. So notice we're reabsorbed sodium chloride kind of everywhere, right, except for the descending limb, which is only permeable to water, right? We can remove water pretty much everywhere. In the proximal convoluted tubules, remember the amount of water and the amount of sodium chloride that are leaving are going to be isoosmotic, right? They don't have the ability to concentrate or dilute whatever they are removing. So you'll get equal numbers of this. And really, it's the water following the sodium chloride, right, in those instances. So that, you know, the amount of water that escapes the filtrate in the two convoluted tubules, proximal and distal, is going to be equal to the amount of solute that's reabsorbed. Remember, the proximal convoluted tubule reabsorbs all organic nutrients. So 100%, or at least that's what we're going to say. We're simplifying it, right? But 100% of nutrients, pretty much, and vitamins and things that are organic and important to the body, like glucose, amino acids, and whatnot, are going to be reabsorbed, so they should not show up in the urine at all. The lute produces a hypertonic medulla, and then the collecting duct is going to fine-tune how much water we're going to release. Now, you can imagine if our collecting duct becomes tolerant or doesn't recognize hormones, that you will start producing a ton of urine, right? I know 20% or 10% of the filtrate doesn't seem a lot, but 10% of 180 liters is a lot, right? I mean, so it could be very dangerous, actually, if that happens. It's very rare. But if you have high blood glucose, you can produce a lot of urine. Because if glucose ends up in the filtrate, what's that going to do? It's going to add more solute, isn't it? You keep water, right, in the nephron, which we don't want to do, and it'll be harder to concentrate the urine, right? So you can produce, when somebody has uncontrolled diabetes, a lot of times they are urinating a lot, right? They're producing a lot of urine. So let's talk about how we control the collecting duct. Okay, you guys are all good with how the collecting duct works and what the loop's job is, right? ADH is going to be our hormone. ADH stands for anti-diuretic hormone, or going against producing urine, right? So ADH is what we call it, or it also has another name. It's called vasopressin. So vaso Vasopressin is involved with blood pressure, right? Vasopressin. So we're going to do a pretty cool lab. I don't know when we're going to do it. Maybe we'll do it next week or the week after. I haven't decided yet. We're going to take frogs and inject them with ADH on purpose. You guys get to do it. We'll inject it into their thigh muscle. It's kind of a fun lab. And then frogs, they absorb water through their skin. So instead of collecting ducts being sensitive to water or ADH, their skin is. And so they will gain weight, and it's kind of fun. So we'll have a control, negative, positive control, and then we'll have an ADH frog, and we'll measure them throughout the lab. And so they'll be jumping all over the place. It's my favorite lab. And then we'll collect a bunch of data from that and be able to write a full lab report. So we'll get a little exposure to what we call vasopressin. And vasopressin is universal pretty much across terrestrial vertebrates. They use it to absorb water, either through the skin or the kidneys. So depending on whether you're an amphibian, mammal, or reptile. So the way this works is this is pretty much a negative feedback loop, isn't it? Right? I mean, when you look at this. So our control center is up here in the hypothalamus. Right? So this is the part of the brain. When we get to the endocrine system, what we're going to learn are there are some nerves up here in the brain that regulate osmolarity. That's their job. And the interesting thing about the hypothalamus is it's one of the few parts of the brain that actually is in contact with the blood supply. It can actually do some blood measurement. It can actually monitor, right, what's going on in the blood. And so there are osmometers here, or osmoreceptors in the brain that can sense an increase in blood osmolarity. Okay, so we know that our control condition here is 300 milliosmoles, right? If it goes up to 350, 375, you're dehydrated, right? You're not drinking. That stimulus is going to stimulate these nerves in the hypothalamus, which will control the secretion of an outgrowth of the brain called the pituitary gland. Okay, and the pituitary gland here, the part that we're interested in the pituitary in this particular instance is the posterior pituitary gland, which releases oxytocin or ADH. All right, and here we're going to just stick with ADH. So when we stimulate these hypothalamic nerves, they will release ADH, which will travel right from the hypothalamus, as we'll see, and we'll learn the mechanism later, to stimulate the posterior pituitary to release ADH into the blood. And it travels through the circulation, because it's a hormone, right, to the collecting duct, and it causes this, water reabsorption, which will further prevent a decrease, or will drop, right, the osmolarity, and it causes tremendous thirst also. And so you'll drink water, you'll reabsorb it, you'll produce concentrated urine, and your blood volume will go up, right? Or your water content will go up. Now notice we're not absorbing sodium chloride here, we're just absorbing water. So there is another hormone that kind of helps with this to prevent too much dilution of our blood, right? We don't want to just add a bunch of water to the blood. There's another one we'll talk about. It's called aldosterone that will also be released here, because ADH will also cause an increased release in aldosterone, which I'll talk about when we get there. All right, but this is ADH, and this is going to be stimulated by an increase in osmolarity, and it works on the collecting ducts to cause water reabsorption from the collecting ducts, so we'll produce a concentrated urine when ADH is present. Now, if ADH is absent, you produce dilute urine. So, a couple things that we ingest that inhibit ADH. Caffeine. Have you ever noticed when you drink a lot of caffeine, you got to pee? Or maybe an hour later. Alcohol. I used to call it breaking the seal back in the day. Right? Alcohol inhibits ADH and causes dehydration. It's one of the main mechanisms for a hangover if you drink too much. So there's a couple things that can inhibit ADH release. Now, what does ADH cause to happen in the collecting ducts? And this is a cool little study that your textbook has that shows the number of aquaporins in the apical membranes of the epithelium that lines the collecting duct. And so notice pre-ADH, upon ADH injection, look at the number of aquaporins. I mean, it's very significant, right? It's statistically significant, big time. And even in the post-ADH, you have a little more, then it's really not statistically significant between pre and post-ADH, but when ADH is present, aquaporin number goes way up, right? In the collecting duct. And along with that, notice what they also showed is that the epithelium is very permeable to water, right? So notice pre-ADH, ADH, post-ADH, we have a lot of water, right? Permeability. Notice that water permeability is still around post-ADH. And that's because you're inserting all of these water channels and it takes time to remove them, right? Out of the membrane. And so for a period of time after ADH stimulation, you still have decently permeable epithelium to water reabsorption. Now, the molecular mechanism has been worked out, and it's a self-signaling mechanism. So why wouldn't I teach you that, right? Since we're talking about it. And so ADH binds to what are called vasopressin 2, V2 receptors. Vasopressin, right? Another name for it. And it's a G-coupled adenylate cyclase receptor, right? And you should be like, oh, that's cyclic AMP, right? And that's exactly what you see here. So notice your ADH comes in, binds to the V2 receptor. This is on the basal side. Notice this is the blood side, right? This is the apical side. This is the filtrate side over here, right? So that we're all in the right mind frame. This is a tight junction up here. This is a tight junction down here, right? ADH is traveling through the blood, leaves the blood, binds to its receptor on the epithelium of the collecting duct, stimulates or changes the shape of the G-coupled protein, which pops GDP off and adds GTP, right, to the alpha subunit, which then will turn on AC, adenylate cyclase, and you get an increase in CAMP. All right, and CAMP, remember, always is going to turn on protein kinase A, all right, or PKA. And protein kinase A does a couple of different things here. What it does is it is going to go out and phosphorylate vesicles that have the AQP2, aquaporin 2 protein in them. Okay? These are pre-made. So these cells are always producing a steady flow of aquaporins, and they are packaged during their packaging in the ER and as they go through the Golgi into the membrane of the vesicles. Okay, notice that. And this is pretty cool. The protein kinase A actually directly phosphorylates them. So it comes along and directly phosphorylates them. Once they're phosphorylated, as you can see the P's sticking off of here, then the microtubules can recognize them. Now, I always say that, right? But what really recognize them is the motor protein, right? And so the dynein is one of our motor proteins. The dynein can now bind to the vesicle and walk it, right? Walk it to the surface. And once we walk it to the surface, the vesicle becomes part of the apical membrane and inserts the aquaporins in it. And now water will flow down its concentration gradient, right? Which is going to be much higher in the filtrate than the medullary space because of the loop of Henle, correct? And so water will flow from the filtrate through the aquaporins and then out over here. You might want, and what's not on this figure, is aquaporin 1 is located in the basal membrane, all right? So AQP1, named based on when they were found or described, is actually found on the basal membrane. So water just, flows through, right? Back into the blood supply. And what that'll ultimately do as urine passes down the collecting duct is it'll concentrate, right? Concentrate or filtrate because we removed water. Also, what I want you to recognize here is that protein kinase A also can enter the nucleus. You see this? And activate this protein here which is called CREB, which is basically a protein that's called cyclic AMP receptor element B. That's where this name comes from. C is cyclic AMP or CAMP. Receptor element. Any receptor element is a transcription factor. Okay? You guys remember what transcription is, right? A transcription factor turns on transcription. Okay? Or it can turn it off too. But in this instance, we're talking about an enhancer here. Right? So it's going to turn on transcription. And so what protein kinase A does is it comes along here and phosphorylates these proteins that are being produced here, right? Here's the gene, here's the protein here, the cyclic AMP receptor element B. Once it's phosphorylated, it will then bind to the promoter of AP2, which is aquaporin 2, the aquaporin 2 gene, turning that gene on so that you can do gene expression, right? And so you'll produce more of these, that is, you'll increase the synthesis, right, of your aquaporins because you're using them, right? So you want to replace what you're using. And so protein kinase A not only phosphorylates the vesicles, but it also turns on a receptor element that then will turn on aquaporin 2 transcription. Once ADH disappears from this release, that is, once our blood volume comes up and our hydration of the blood goes up, you can imagine the hypothalamus stops stimulating the pituitary to release ADH, right? And then this will start slowing down. And when it starts slowing down, we will recycle at the membrane. We'll pull these aquaporins off because it's the phosphate. It's pretty interesting. At least this is what they hypothesize. This phosphate is going to be removed after a certain amount of time. And once it's removed, then you will pull them out. You'll pull them back out. So the phosphate not only signals the insertion, but kind of has this stopwatch effect where it's like, all right, it's phosphorylated for so long before it's removed, and then you pull the aquaporins back out. You can store them in a vesicle, reuse them a couple times, and if they start to break down, we can degrade them, right? So we can do some membrane recycling with the endomembrane system. Also, another enzyme here, phosphodiesterase, does the opposite of adenylate cyclase. It will break CAMP down into AMP, and so that eventually when ADH disappears, we have to remove the CAMP, right? We have to remove the CAMP, so that's what phosphodiesterase does. Once ADH is gone. So that's how we stimulate these aquaporin insertions. So now you know why we went through all that, right? Questions? Okay, let's talk about the other hormones. So if you just relied on ADH, you would dilute the blood. I mean, it could ultimately cause dilution of the blood. To be honest with you, in reality, most mammals that are terrestrial like us, we release bursts of ADH all day long, just so that we can kind of maintain a certain blood osmolarity, right? You can increase more release or decrease release of it depending on how much water you're drinking, correct? Or how much fluid you're drinking. Or if you drink caffeine or alcohol, it can inhibit ADH, right? But we don't want to dilute the blood too much, right? From urine. So we have another hormone here that is called aldosterone, okay? So aldosterone. So aldosterone is going to be found in the adrenal glands. Do you guys know where the adrenal glands are? You may not.
Top of the kidneys.
You're good. Right on top of the kidneys, which is good because it's the kidneys that are going to stimulate them, right? And so aldosterone is released by the adrenal glands, which sit on top of the kidneys. Yeah, they sit right on top. And the mechanism is pretty cool. it's pretty complex, but it's pretty neat what it does. And so now the kidney here is actually our sensor, our control center, and all of that. It's all kind of built into the kidney itself. So how does this work? How does the kidney sense when there is a drop in osmolarity, right? There are a couple different ways that the kidney can do this. Now, if you remember, we talked about our anatomy here. This is our renal corpuscle, right? So we have Bowman's capsule. On the outside here, we have the the parietal layer of Bowman's capsule, right, which is a simple squamous epithelium. We have Bowman's space, and then this is the glomerulus, right, which are the capillaries, and then the visceral podocytes, right, the podocytes that form the filter. Here's our afferent arteriole coming in, right, which will become the capillaries, the glomerular capillaries, and then they will drain out the efferent arteriole to what we call the peritubular capillaries that surround, right, the renal tubule. This is a great drawing of that, right? So here we got the afferent coming in. Here is the glomerulus. Here's the efferent leaving. What's really neat about this is that as the loop, as you can see here, as we enter the loop and we drop down, right, we're then going to come back up. And notice what happens here. The distal convoluted tubule is organized in such a way, because of the loop, that it comes in really close proximity to the blood supply. And it actually passes between the afferent and efferent arteriole. Do you see that? And so it's basically, if this, my fist, is the renal corpuscle, it's pushed against the renal corpuscle, right? Right next to the blood vessels and the glomerulus. So for a histologist, that's not coincidental. It happens in all of them. This is the normal organization. So, and if you look at this in cross-section, coming up out at me is going to be the distal convoluted tubule. Okay? Not only does the distal convoluted tubule come out at me, but at the level of the afferent arteriole and on the side, of the same side, right, of the afferent arteriole, the epithelium becomes tall. It becomes really tall. Histologists notice these little things. And this taller epithelium has a name. It's called the macula densa. Right? It has a name called the macula densa. Whereas the rest of the distal convoluted tubule is mostly like cuboidal epithelium. And so this is a picture from a rat here, where I got a really nice picture here of these cells. See how these cells are right there, right? They're all kind of sandwiched right here. You can't see the blood supply coming in, but you can see these cells here with the big round nuclei. That's the macula densa. And then these cells right here that are kind of flatter, darker nuclei, they're called juxtamedullary cells, right? Juxtamedullary cells. Juxtaglomerular cells. In some texts they're called juxtamedullary cells, but juxtaglomerular means right next to the glomerulus, right? So you got, and they travel in here. You can see they're usually kind of, what do they call them? They call them spindle-shaped or football-shaped cells. And they typically have a dark nucleus like this. And they're going to be sitting basically between the glomerulus and the macula densa. Okay? So this layering has anatomical significance, architectural significance, because it is part of the monitoring system that will eventually cause the release of aldosterone if they're stimulated. Okay? There are also smooth muscle cells that are found in the wall of the afferent arteriole. Like any arteriole, they have smooth muscle that can squeeze down on the blood vessel and relax on the blood vessel, right? To increase and decrease blood pressure. Now, as blood enters the afferent arteriole, the blood is going to do this. It's going to stretch, right? As it enters with each heartbeat, as it enters the glomerulus, and this stretch can be monitored, right? So too much stretch means high blood pressure. Too little stretch means low blood pressure. Normal stretch is normal blood pressure, right? And so these juxtaglomerular cells can pick that up. They can pick up the stretch, okay? And if there's too little stretch coming in, that means blood volume is low, osmolarity is high, right? And they will release, these juxtaglomerular cells will release renin, all right? Sometimes you'll hear it called renin. And some books call it a hormone. In reality, it's an enzyme, right? They release renin into the blood. So, obviously, if there's too much stretch, right, that means you got high blood volume, high blood pressure, you won't release renin. But if your stretch drops and it's not enough, that is the blood pressure coming into the afferent arteriole is not high enough, then you release renin into the blood supply. Now, what renin does is it's an enzyme, and its substrate is this protein here that actually is called angiotensinogen. See? All right, angiotensinogen. So angiotensinogen is a liver protein. So your liver produces angiotensinogen constantly. It's just going to produce it, drop it into the blood at a constant rate. So you have a constant concentration of angiotensinogen traveling through the blood supply. Now what renin does is it binds to this. Whenever you see ogen on the end of a protein, it's inactive. Okay? It's an inactive protein. So, angiotensinogen is traveling. If I sense a drop in blood pressure, these juxtaglomerular cells will release renin. Renin will bind to the angiotensinogen and basically cleave off a part of the protein. So, this protein, angiotensinogen, is going to have an end to it, some amino acids on the end of it, that prevent it from folding up. All right? And a lot of proteins are produced like this, where they're produced in kind of in an inactive state. And the easiest way to do that is to prevent protein folding, because we know the three-dimensional shape is what allows them to do what they do, right? So if I add some amino acids on the end of them that are not significant, they simply prevent the protein from folding up right, and the protein doesn't work. And so what renin does is it comes in and it cleaves that off. Right? That's what renin does. It cleaves off those amino acids on the end of the protein. And now what can happen? My protein can fold, right? It can fold normally. And when it does that, it will produce this protein called angiotensin 1, right? Which tells you it's going to change shape, right? So angiotensin 1, the first thing that I want you to know about angiotensin 1 is it is a fantastic vasoconstrictor. So vasoconstrictor. So vaso means blood, right? Blood vessel. Constrictor means it will constrict the arterioles of the body, and that will back blood up into the arterial system. And it will increase blood pressure. So increased vasoconstriction leads to increased blood pressure. Right? I just told you blood pressure was too low. So it's like an immediate effect, right? So very potent angiotensin 1 vasoconstrictor. Matter of fact, it starts working immediately and causes vasoconstriction because it's traveling right through the blood. It causes kind of vasoconstriction in the efferent arteriole and backs blood up, which increases blood pressure immediately, right? Which will help with filtration, won't it? Because ultimately, that's what we're doing. Remember, I kind of mentioned this. You have a flow rate to the filtrate. And if blood pressure drops, filtration drops, filtration flow drops, right? And so does filtrate movement drops. Urine production drops, doesn't it? Right? So you kind of want this constant flow rate, constant filtration, constant movement or flow through the nephron. And if we have slow movement, that means our filtration pressures drop. Remember that? And so we're not pushing fluid as much. And so if I constrict the efferent arterial smooth muscle because of angiotensin 1, then blood's going to back up and going to increase filtration rate, isn't it? Right? But it also causes systemic vasoconstriction, which can increase blood pressure everywhere once it travels past the efferent arteriole. Now, angiotensin 1 circulates. once we've activated. And when it goes through the lungs, it's going to meet another protein called ACE, angiotensin or angiotensinogen converting enzyme. You guys probably remember hearing of ACE because the ACE receptor or the ACE protein allowed what to enter lung tissue and shut down the whole school, shut down the whole U.S., shut down the world. It's what COVID virus binds to, right? So isn't that weird? This is not only an enzyme, it's also a receptor, and is what COVID-19 used to damage lung tissue. Okay. Now, what ACE normally does, it's got lots of jobs, but one of the jobs that it does in the lung tissue is it takes angiotensin 1. See if I can find another color here. No, Chuck. Oh, we got green, right? So we'll put some other enzymes down, or some other amino acids down here. ACE cleaves off the other end of the protein and allows it to further fold into angiotensin 2, which has a specific shape. And notice what angiotensin 2 does. Again, another vasoconstrictor, right? Just like angiotensin 1. Promotes thirst. So just like ADH, it's when in the hypothalamus, it also stimulates thirst behavior. Stimulates vasopressin secretion. Ooh, vasopressin's ADH, isn't it? Right? And then causes the absorption of sodium chloride. And guess what? That's going to be in the collecting duct. And it does that through this hormone called aldosterone. So a stimulation of renin causes angiotensinogen conversion to angiotensin 1. In the lungs, we take 1 and convert it to 2. And 2 binds to cells in the adrenal glands and causes aldosterone release. Aldosterone travels to the collecting duct where it meets its receptor. Okay? Holy moly, how fun. You don't look like this is as fun as I think it is. So far, so good, right? Now, there's even a cooler mechanism that, of course, I got geeked out on, right? And I wanted to teach you. So there's another way we can cause renin release. The juxtaglomerular cells obviously are the ones that release the renin, and we know they can kind of monitor stretch, right? Monitor stretch. But think about this. If blood pressure drops to the afferent arterial, overall filtration pressure drops and leakage into the nephron drops, right? And so your filtrate flow slows down, right? And you might think, well, why is that a big deal? You're not producing a lot of urine. Okay, great. But if everything slows down, then reabsorption is going to increase, right? Because there's more contact time. The more contact time, the more reabsorption, the more secretion you get. You want a flow rate that's consistent so secretion and reabsorption is constant. You don't want to increase reabsorption. So think about it. If everything slows down and I'm passing through the whole nephron slowly, when I get to the distal convoluted tubule, I'm going to have reabsorbed too much sodium chloride, right? Because I allowed particularly, right, this fluid to slow down in the ascending limb. And remember that multiplier? It's going to remove too much sodium chloride because the flow rate is slow. The macula densa can sense that. It can sense a drop in sodium chloride below what's normal, right? It's controlled condition because flow rate is too slow. When it does that, it stimulates the juxtaglomerular cells to release renin by sensing sodium chloride. Not a direct sense of blood pressure, but by sensing flow rate through sodium chloride concentration. And how cool is that? That's a really cool mechanism. So you got two ways here to kind of cause the release of renin through the juxtaglomerular cells, either through less stretch of the blood vessel or less sodium chloride concentration than normal in the filtrate that's passing along the macula densa. And then you guys are, right, you're budding physiologists, and the question that comes to your head is, how? Right? It probably isn't. But for me it is, right? So how do they do that? It's pretty simple how they do it, right? This is a new figure, right? And I'll upload it, the updated PowerPoint, because I wanted to go ahead and show you this. I usually draw it, but I didn't want to draw it today. I was too lazy. My drawings usually are poor. So this is kind of a blow up of that area, right? So here's the afferent arteriole over here. Here are your juxtaglomerular cells, right? Juxtaglomerular cells. Here is your macula densa. So here's the filtrate right here. Macula densa cells, and then the afferent arteriole. So local baro reflex. Local baro. So baro means pressure, mechano pressure. So these local baro receptors are what sense the pressure, right? And if there's a drop in pressure, they, notice the arrow, stimulates the juxtaglomerular cells to release renin. Okay? Now don't worry, we're not going to learn everything on it. Okay? But that's kind of what we talked about before. The way they do it is by increasing calcium. Ooh, wow, that's real surprising, right? They cause calcium channels to open. And those calcium channels are going to allow calcium into these cells, and that's going to cause the release of renin, right, by lighting up the vesicles that are holding it, right? Renin is a protein, obviously, right? So stretch causes calcium to increase in the juxtaglomerular cells, or they're called renal granular cells. That's actually what I've always called them, because they're under a microscope look granular, and it's the vesicles of renin that they're holding. That'll liberate them if there's not enough stretch. Now let's come over here and look at these guys. This is the last thing I'll torture you with today. Okay? So here is your macula densa cell, and wow, look at that. It's got that same protein in it, that same symporter. Sodium, chloride, potassium, symporter. It's not on here, but there is a sodium-potassium pump over here, right? Oh my gosh, there's hydrogen, proton, and sodium, so that means carbonic anhydrase. I mean, have you noticed the common theme? All the cells kind of use the same proteins. And you can imagine if flow rate's normal, let's say you've got normal blood pressure, there's a normal amount of sodium entering, right? If the filtrate is slow, when they get here, there's not a lot of sodium coming in. The filtrate's moving slowly. There's just not a lot to come in. There's not a large concentration out here to drive it in, no matter how low this is. And so sodium and chloride and potassium levels drop because the filtrate is moving too slowly. Okay? The cell senses that. It can sense that decrease in sodium coming in, and that'll turn on this enzyme. Okay? This enzyme, which we'll talk about later too, it's called COX. There's COX-1, COX-2, COX-3. It's called cyclooxygenase. Cyclooxygenase. This one is cyclooxygenase 2. Right? So cyclo, oxygen, and then add ace to the end of it. Right? Cyclooxygenase. And what this is a membrane-bound protein. They're not quite sure, and that's why I'm not teaching you this, how sodium coming in turns it on, right? There are some hypotheses out there, but I haven't found one that I'm real keen on yet. So, but obviously the flow of less sodium turns on COX-2, and what COX does is it's going to do something like phospholipase. It's going to modify phospholipids. It'll take a phospholipid and turn it into a prostaglandin. You guys ever heard of a prostaglandin before? So a prostaglandin So the one here is prostaglandin PGE, prostaglandin E2. Right? There are a whole bunch of different prostaglandins. A prostaglandin is one of the fatty acid tails. So you cleave off the fatty acid tail, you might modify one of those fatty acid tails. A prostaglandin is a long carbon chain. It is a really potent inflammatory or ligand, right? And so it usually is involved with inflammation. But here, what happens is the prostaglandin 2, which is produced by COX, will travel over to the renin cells and cause an increase in calcium, right? And it's a phospholipid, so it can go ahead and enter right into the cell, right? It's part of a phospholipid tail, so it's pretty hydrophobic. It can travel right into the cell and cause calcium to increase. When calcium increases, guess what? We release renin, right? The COX enzymes, you know what they're involved in a lot is pain and inflammation. So a lot of the inflammatory response that happens somewhere where you're hurting, like a cut, is driven by inflammation, right? And that is driven by this COX enzyme. So things like Advil, aspirin, acetaminophen, they block that. They block that enzyme, and then they block pain. Kind of how they work. So that's kind of how this works. So it's kind of a metabolic sensing in the sense of it's really a flow rate. It's a flow thing, right? It's a flow rate sensor through sodium chloride production. Another thing that's turned on here, not only COX is turned on, but so is NOS. All right, and we're going to talk about this enzyme also. NOS stands for nitric oxide synthase. Nitric oxide synthase. So this decrease in sodium turns on NOS. And NOS produces nitric oxide, if you remember, right? Nitric oxide. And look what nitric oxide does. It's going to travel over and into these juxtaglomerular cells and cause the release of renin, right? And the question I'm going to ask you to kind of think about, and we'll talk about it at the beginning of the lecture tomorrow, is how does NOS do that? What does nitric oxide do? You already know. And I'll let you tell me tomorrow, right? All you got to do is go back. If you don't remember, go back and look at the second messenger. Remember that? And that was lecture 2 or 3. The second messenger slide that has, cyclic AMP, cyclic GMP, blah, blah, blah. And look for NO, nitric oxide, and see what it stimulates. And that's exactly what it's going to do in these juxtaglomerular cells. It's going to bind to an enzyme and turn it on. Okay? How cool. Pretty cool, right? All right, I'll stop there. All right, we'll start back up here and talk about NOS and maybe PGE2 a little bit. Under this figure, I'm going to put this online today. There's also a figure legend. This is from a paper. And this is showing you some of the immunocytochemistry they did, but it will give you the names of all these things if you really want to know them, right? We'll come back to this figure and then start on this one tomorrow. So look up what nitric oxide does. And you'll see it does that, or you're going to see it does that in these cells. All right, I will also put this up tonight, if it works. All right?